but also their state of health at death. At the British Museum, we've been using a very powerful new CT scan, and that allows us to capture both the thinner elements, the delicate skin and textiles, equally as well as the more dense elements such as the skeleton. If you compare it to the data we captured 10 years.
For more than a century, the dead of Egypt were treated like monuments, wrapped in linen, sealed in stone, catalogued behind glass. They belonged to history, not to conversation. Their faces had been reconstructed, their tombs had been mapped, their dynasties had been named, and for a long time, it seemed that ancient Egypt had already told the world everything it was ever going to say. Then the genetic era arrived, and suddenly, silence was no longer enough. Because when scientists began searching for DNA inside Egyptian remains, the results did not come easily. The heat was wrong, the chemistry was wrong, the passage of time was wrong. Egypt, of all places, preserved bodies magnificently, but often destroyed the very molecules modern labs needed most. Again and again, researchers were warned that any signal they found might be too damaged, too contaminated, too mixed with the fingerprints of bacteria, handlers, soil, embalming compounds, and time itself to trust. The kingdom of mummies was also a kingdom of noise. And yet, pieces of code began to emerge. Fragments, traces, reeds that aligned, histories hidden in bone. Evidence that some Egyptian individuals carried ancestry stretching across North Africa and into the Eastern Fertile Crescent. Evidence that microbes and pathogens also lingered in the dead. Evidence that tombs were not sealed time capsules, but biological battlefields. And that was the moment the story split in two. On one side stood the laboratory, cautious, technical, suspicious of sensational claims. On the other stood the internet, where one phrase could ignite everything: strange DNA, not human. But what had actually been found in those ancient remains? A biological impossibility? A misunderstanding of contaminated data? Or something far more unsettling? The discovery that the greatest mystery was never in the tomb at all, but in the way the modern world reads the dead. Ancient Egyptian human remains have been the subject of genetic research for decades, but successful recovery of authentic DNA has been notoriously difficult. Heat, humidity in some burial contexts, chemical treatments associated with mummification, and extensive handling after excavation all increase degradation and contamination risk. That difficulty is not hypothetical. It is one of the central technical facts of mummy genetics. In 2017, a landmark study reported the recovery of 90 ancient Egyptian mitochondrial genomes and genome-wide data from 3 individuals from the site of Abusir el Malek in Middle Egypt. The study did not report non-human ancestry. It reported human genetic data and argued that the sampled ancient population showed affinities with ancient Near Eastern populations, while also warning that one site cannot represent all of ancient Egypt. In 2025, researchers reported the first whole genome from an Old Kingdom Egyptian individual, a man buried at Nuweiret, roughly 4,500 years ago. The results suggested that most of his ancestry aligned with North African Neolithic populations, with roughly 20% linked to the Eastern Fertile Crescent. Again, this was a fully human genome, not an anomalous, non-human lineage. There is also confirmed evidence that Egyptian mummified remains can preserve non-human DNA, but in a very specific sense. Metagenomic work has identified microbial and pathogen DNA, including bacteria and hepatitis B virus, in mummified individuals. In other words, non-human DNA in a mummy sample can be real without meaning anything supernatural or biologically impossible. It may reflect microbiomes, disease organisms, decomposition, environmental intrusion, or later contamination. Scholars continue to debate how representative current samples really are. One cemetery does not equal one civilization. One individual does not define a kingdom that lasted millennia. Genetic data from elite tombs, provincial burials, pre-dynastic graves, and later period mummies may tell different stories. This is why serious researchers speak in cautious, site-specific terms, rather than sweeping declarations about the Egyptians as a single, unchanging population. There has also been long-running debate about older royal DNA work, including the 2010 Tutankhamun Family Project. That study claimed to establish family relationships among royal mummies and identify evidence consistent with malaria in some individuals. But ancient DNA work on royals has historically drawn intense scrutiny because contamination control, reproducibility, and public interpretation are extraordinarily difficult in such high-profile cases. This is where the story often leaves science and enters mythology. Online narratives sometimes take any unmapped, damaged, or microbial sequence and reframe it as proof of an unknown species, hybrid bloodline, or hidden chapter of history. There is no verified mainstream study showing that DNA recovered from an ancient Egyptian tomb belongs to a non-human intelligent being. That claim remains speculation, not established fact. And yet the speculation persists because Egypt has always attracted a certain kind of projection. When the evidence is incomplete, imagination rushes in. The anomaly begins with a contradiction. Egypt is one of the most visually preserved ancient civilizations on Earth. Linen survives, coffins survive, hair survives, resin survives, faces survive. Yet the one thing modern science most wants, robust nuclear DNA, often does not survive well at all. That alone is enough to create suspicion. Because when laboratories sequence an ancient sample, they do not receive a pristine strand labeled with a name and a dynasty. They receive a storm of fragments. Some come from the original individual. Some come from bacteria that colonized the body after death. Some come from microbes that lived in or on the person during life. Some may come from excavation, storage, museum handling, or modern laboratories. In poorly preserved samples, the majority of reads may not map cleanly to the human genome at all. To a geneticist, that is a technical problem. To the public, it can sound like an impossible revelation. And that is where the phrase, "it's not human," becomes dangerous. Because in one sense, part of the DNA in a tomb sample may indeed be non-human. It may be bacterial, viral, environmental. It may belong to ancient pathogens, oral microbes, decomposers, or trace biological material from the burial environment. But that does not mean the tomb contained a non-human person. It means ancient remains are chemically and biologically complicated. The deeper anomaly is more subtle and more interesting. Why did some samples fail for decades, while others suddenly yield results? Why did one Old Kingdom skeleton buried in a ceramic vessel preserve enough DNA for a whole genome, when so many mummies, richer and more famous, frustrated researchers for years? Why do certain burials speak more clearly than others? Because sometimes, the greatest mystery is not what the DNA says. It is why the DNA survived long enough to say anything at all. The breakthrough did not come from a single shocking sequence. It came from method. Ancient DNA science changed when researchers stopped treating old remains like ordinary forensic samples and started treating them like molecular crime scenes. Clean rooms, protective suits, independent replication, damage pattern authentication, targeted enrichment, ultra-high throughput sequencing, statistical models built to separate authentic ancient fragments from modern contamination. That is how Egyptian DNA research finally moved from rumor to reproducible evidence. Then came the next leap, metagenomics. Instead of looking only for human DNA, scientists began sequencing everything in a sample. That changed the question from, can this mummy yield a genome? To, what entire biological world is preserved here? Suddenly, tomb samples could reveal pathogens, oral bacteria, decomposition signals, and traces of ancient disease. Some of the strange, non-human DNA was not strange at all. It was the microbial afterlife of the tomb. And then came the 2025 breakthrough, the first full genome from ancient Egypt. Not from a golden pharaoh in a perfect coffin, but from a man buried before standard artificial mummification had become the norm. His preservation may have depended less on royal treatment and more on a rare chain of environmental luck. His genome did not reveal anything non-human. What it revealed was something historians consider more valuable: evidence that ancient Egypt was connected to wider human movements across regions long suspected to be in contact. AI can help sort immense sequencing datasets, flag contamination patterns, compare fragments against massive genomic databases, and classify likely microbial signatures faster than older manual workflows. In future studies, machine learning may become increasingly useful in distinguishing ancient damage from modern intrusion. But AI cannot magically transform bad DNA into good evidence. It cannot invent ancestry where the fragments do not support it, and it cannot by itself declare a sample not human. That conclusion still depends on biological context, replication, controls, and human scientific judgment. That is the real breakthrough of the modern era. Not louder claims, but better filters. But breakthroughs do not end controversy. They deepen it. The moment ancient Egyptian DNA enters public conversation, three forces collide: science, identity, and spectacle. Science wants caution. It wants enough samples to rule out coincidence. It wants better geographic coverage. It wants comparisons grounded in archaeology rather than ideology. It wants the audience to understand that one cemetery in Middle Egypt does not stand in for an entire civilization spanning thousands of years. Science wants caution. It wants enough samples to rule out coincidence. It wants better geographic coverage. It wants comparisons grounded in archaeology rather than ideology. It wants the audience to understand that one cemetery in Middle Egypt does not stand in for an entire civilization spanning thousands of years. Identity wants certainty. Ancient Egypt sits at the intersection of Africa, the Mediterranean, and Southwest Asia, and modern people often approach it carrying contemporary political arguments about race, migration, heritage, and ownership. Every genome becomes tempting to weaponize. Every caveat becomes inconvenient. Every nuance becomes vulnerable to headlines that are cleaner, sharper, and less true. And spectacle wants a twist. A fully human genome with mixed regional ancestry is historically important. But to a sensational algorithm, it is not enough. The phrase microbial DNA found in mummy sample does not spread like scientists found DNA that wasn't human. The latter is explosive because it seems to promise forbidden knowledge. The former is merely science. Institutional hesitation grows in that atmosphere, and not always for sinister reasons. Sampling ancient remains can be destructive. Permissions matter. Museum ethics matter. National heritage authorities matter. Researchers know that once a claim escapes into the public sphere, it may never return to context. That caution can look, from the outside, like secrecy. Critics may allege gatekeeping, but often it is simply the cost of handling fragile evidence in a world addicted to overstatement. There is also the stubborn problem of reproducibility. Ancient DNA is not like turning on a machine and reading a barcode. Different labs, different extraction methods, different preservation conditions, and different contamination histories can shape results. That is precisely why the field has matured around authentication standards rather than dramatic first impressions. The stakes rise further because every new success changes expectations. Once one full genome appears, the world wants 10. Once one pot burial yields evidence, the world wants a royal tomb open next. Once one ancient microbiome is recovered, the public begins asking whether every unexplained sequence is a hidden species. And this is where careful science becomes almost tragic in tone. Because the researchers are not standing at the edge of a fantasy. They are standing at the edge of a dataset, trying to protect truth from desire. The strongest conventional explanation is also the one best supported by evidence. Claims of non-human DNA in ancient Egyptian tombs are usually misunderstandings of microbial DNA, pathogen DNA, environmental contamination, or unmapped degraded fragments. Ancient remains are biological ecosystems. Sequencing them often reveals bacteria, viruses, and other non-human material alongside the damaged human signal. That is expected, not impossible. Under this interpretation, the mystery is not alien or superhuman. It is technical. The public is hearing a laboratory description without laboratory literacy. A more adventurous, but still plausible interpretation is that some ancient tomb datasets may preserve biological signatures that modern databases do not classify well yet. Not because they come from non-human intelligences, but because ancient microbial ecologies, burial chemistry, pathogen evolution, and degraded fragment patterns can be poorly represented in present-day reference libraries. In that version of the story, the anomaly is real, but it belongs to ancient disease environments, mummification materials, burial conditions, or lost microbial communities. The DNA looks strange, not because it is beyond Earth, but because the past does not always fit neatly inside modern databases. The skeptical position is harsher. It says there is no genuine anomaly here at all, only the inflation of ordinary ancient DNA complications into pseudo-mystery. It argues that the title itself is a symptom of a broader problem, the tendency to treat Egypt as less human precisely because its achievements feel too monumental, too remote, too symbolically overloaded for modern culture to leave alone. From that view, the phrase, "it's not human," tells the audience more about modern appetite than ancient evidence. From that view, the phrase, "it's not human," tells the audience more about modern appetite than ancient evidence. There is also a fourth interpretation worth mentioning. When genetics reveals ancestry links to other regions such as the Eastern Fertile Crescent, some audiences mishear non-local as non-human. That leap is false, but emotionally revealing. Human migration has always unsettled simple origin stories. A genome that complicates identity may get exaggerated into one that breaks biology, and so the audience is left with three concentric mysteries: what the sample contains, how the data were framed, and why the framing became more powerful than the science. This matters because the dead of Egypt are not abstractions. Behind every sequence read is a person who once ate, worked, aged, suffered, and was buried by others who believed memory could survive stone. The 2025 genome did not come from a legendary ruler. It came from an older man, possibly a potter, whose bones preserved traces of labor as clearly as they preserve traces of ancestry. That detail changes the emotional scale of the story. Ancient DNA is no longer only about dynasties. It is about recovering ordinary human presence from extraordinary distance. It matters because history is often written by inscriptions, monuments, and elites. Genetics offers another voice, incomplete, fragile, but different. It can confirm migration where texts are silent. It can expose disease where mythology is mute. It can complicate identity where slogans demand purity. And it matters because mystery itself has a moral edge. When modern culture sees something astonishing in the ancient world, it often rushes toward dehumanization. The builders must have had help. The kings must have been hybrids. The tomb must contain something other than human. But perhaps the more unsettling possibility is the opposite, that the past was fully human, and humanity has always been more connected, more sophisticated, and more biologically entangled than simple stories allow. The question, then, is not only what remains in the tomb, it is whether the modern world is prepared to accept a past that is miraculous without being inhuman. So the chamber remains open, but not finished. Scientists have recovered real human genomes from ancient Egypt. They have also recovered real, non-human DNA from the microbial and pathogenic worlds preserved around the dead. What they have not verified is the sensational claim that an ancient Egyptian tomb has yielded a non-human being. And yet the phrase continues to echo because it touches something old and restless in the imagination. Perhaps that is the final irony. The tomb did not have to hide an alien secret to be extraordinary. A fragment of bone, a damaged strand, a potter buried before the pyramids reached their full age, a civilization once thought silent, speaking again through molecules. But one question still refuses to go away. When the next unopened sample from ancient Egypt is finally sequenced, will it reveal a biological impossibility, or will it reveal, once again, how easily mystery begins when evidence is only half understood? What does the audience think: misunderstood, contamination, lost microbial history, or a genuinely deeper anomaly still waiting in the dark?